Dear Chairman, dear colleagues, first of all, I would like to thank the organizing committee for the opportunity to present our data. In this study, we investigated mitochondrial DNA mutations associated with atherosclerosis and elaborated approaches to edit mutated mitochondrial DNA by CRISPR-Cas9 system. Mutations in mitochondrial DNA became a subject of intensive study since they may directly lead to mitochondrial dysfunction, oxidative stress, cell dysfunction, and death. Many mitochondrial DNA mutations were found to be associated with chronic human diseases, including cardiovascular disorders. The hypothetical mechanism of atherogenic effect of mitochondrial DNA mutations may be due to the enhanced production of reactive oxygen species and increased oxidative stress, the development of mitochondrial dysfunction, inflammatory reaction, cell death, and dysfunctional mitophagy. Mitophagy is an important element of mitochondrial quality control. There are three types of mitophagy pathways. This is ubiquitin-dependent pathway, ubiquitin-independent pathway, and alternative pathways. Defective mitophagy causes a pronounced accumulation of damaged mitochondria and excessive inflammation caused by pro-inflammatory cytokines that can contribute to the development of inflammatory and autoimmune diseases. Modified atherogenic LDL form LDL self-associates that penetrate the cell by non-specific phagocytosis. LDL-stimulated phagocytosis activates their pro-inflammatory reaction. Secretion of cytokines leads to increased accumulation of intracellular lipids. If the innate immunity functions normally, the pro-inflammatory reaction resolves rather quickly and the further lipid accumulation does not occur. However, when macrophages contain mitochondrial DNA mutations, their pro-inflammatory response does not rest, but rather intensifies with each repeated pro-inflammatory stimulation. The cause of mitochondrial dysfunction may be associated with the defective mitophagy. Local inflammation in the vascular wall becomes chronic and accompanied by uncontrolled lipid accumulation, giving rise to an atherosclerotic lesion. Another intriguing possibility is that cells may recognize the dysfunctional mitochondrion as a pathogen that presents foreign epitopes, therefore triggering the immune response. This may be a consequence of the bacterial origin of mitochondria. The proposed concept allows speculation that atherogenesis is due to two errors made by the cell of the arterial wall. The first one is that the cell perceives the associates of modified LDL as a pathogen that is taken up by phagocytosis, which causes an inflammatory response and the accumulation of intracellular lipids, which in turn is a trigger of atherogenesis at the cellular level. The second is that due to mutations, the mitochondria becomes dysfunctional, and due to a defect in mitophagy, the cell cannot free itself from these mitochondria and perceives it as a bacterium-like pathogen. This triggers an ongoing inflammatory signaling that may lead to inflammasome activation. Mitochondrial dysfunction may be linked to toll-like receptor and LRP3 signaling, with mitochondria therefore playing a key role in the sterile inflammation. Together, these processes result in chronification of inflammation. Such a development may be characteristic not only of atherosclerosis, but of any pathology associated with the chronification of inflammation. Earlier, we have shown with the sequencing of mitochondrial DNA that several heteroplasmic mitochondrial DNA mutations were associated with atherosclerotic lesions. Heteroplasmin levels of atherosclerosis associated mitochondrial DNA mutations were determined in grossly normal and atherosclerotic segments of morphologically mapped aortic walls. In total, 265 normal and atherosclerotic tissue loci of 5 human aorta samples were investigated. On the left picture, you can see the atherosclerotic burden. The number of dots indicate their atherosclerotic lesion. The absence of dots means normal intima. 1 dot means fatty infiltration, 2 dots fatty streak, 3 dots lipofibrous plaque, and 4 dots fibrous plaque. In the middle picture, you can see mutational burden, where no dots means no mutations, and the increasing number of dots means increasing number of mutations. And the third picture is an overlay of arterial lesion and mutations. So as a result, mutational burden independently can explain about 14% variability of atherosclerotic lesions. The number of heteroplasmic mitochondrial DNA mutations and the integral mitochondrial DNA mutational burden correlated with the extent of atherosclerosis. As you can see on the picture, the largest number of mutations was found in lipofibrous plaque. Similarly, mitochondrial DNA mutations were found in mitochondrial DNA from circulating leukocytes and in patients with atherosclerosis. The heteroplasmin level in human leukocytes was determined by parasequencing method adopted for conditions where both mutant and normal allele were present in the same specimen. We investigated correlations of elucidated mutation with the intermediate sickness, then carotid plaque, coronary heart disease, and acute myocardial infarction. Some mutations have positive correlation, so they were atherogenic, and some mutation had negative correlation, so they were anti-atherogenic. Then to study mitochondrial DNA mutations and their role in atherosclerosis, we established several cell lines with the mitochondrial DNA burden. We used platelet from patients with atherogenic mitochondrial DNA mutations as a mitochondrial donor. We've used this platelet with Ronald THPL1 cells without their own mitochondria and obtain hybrid cells with atherogenic mitochondrial DNA mutations. As a result, we created cell lines carrying the mutation listed in this table. One of these hybrid line was HS-MAM1. And we used this hybrid cell in the current study. So about the mutation, the 15059 mutation in the cytochrome B gene is mainly found in lipofibrous plaques and positively correlates with the age of patients with the coronary atherosclerosis and coronary artery disease. This mutation is a nonsense mutation that causes the formation of a stop codon, resulting in the loss of 276 amino acids. This loss indicated on the picture by red color. This genetic defect may cause oxidative stress in the intima of the human aorta, which can lead to the local occurrence of atherosclerotic lesions. It's also known that this mutation can lead to various diseases, including muscles, nervous system, heart, and kidney disorders. To study the role of this mutation in atherosclerosis, we modified Cas9 plasmid to target mitochondrial cytochrome B gene. For that, we added mitochondrial targeting sites, then modified the Cas9 protein, and optimize the whole system to penetrate mitochondria. Then we tested this vector on THP1 cells, shown by PCR analysis and Sanger sequencing analysis that our vector can produce double-strand breaks in target area of mitochondrial DNA. Then, with the digital PCR analysis, we found that our system can cleave up to 92% of mitochondrial DNA carrying the mutation, and the heteroplasmy level of this mutation was reduced up to 3.7% from 68%. Moreover, we found that some double-strand breaks were repaired by the mechanism of microhomology-mediated end join. The possible matrix for this mechanism was a part of our vector that was delivered to mitochondria together with the RNA complex. This result confirmed the opportunity not only eliminate mutated mitochondrial DNA with the CRISPR-Cas9 system, but also a possibility to create an approach to incorporate a mitochondrial mutation to DNA. And we used this system on our hybrid cells, HS-MAM1. These cells is characterized by defective mitophagy. You can see the results of the measurement of mitophagy on the pictures. So in these cells, it was defected and treated with our system, the mitophagy restored. This result means that mitochondrial DNA mutation can contribute to defective mitophagy. My acknowledgements, my special thanks to my students Vasily Sukhorukov and Victoria Hotina, who participated in all experiments on cells, and Vasily made a huge contribution to the development of the plasmid. Thank you